Okay, folks, today we will talk about protein function. So we've talked about protein structure, and today we're really going to get into a specific example of how structure impacts function. A handful of learning outcomes for the day, some more important than others. We'll understand a bit about both myoglobin and hemoglobin, binding curves, their general structure and function. We'll compare myoglobin to hemoglobin, and then we'll get into the nitty-gritty of today's videos. We'll talk specifically about the R and T structures of hemoglobin, and we'll understand some ways in our body that the structure of hemoglobin changes according to what is present in the environment. Today's Meet a Scientist moment is to Yu. She discovered artemisinin, which is a drug used to treat malaria. It's interesting because this drug is thought to be activated, in red blood cells by the heme. She was the first Chinese woman to ever win a Nobel Prize, and she won the Physiology and Medicine Prize in 2015. So what are myoglobin and hemoglobin? They are proteins that reversibly bind oxygen. So they can bind oxygen, they can release oxygen. Myoglobin was the very first protein ever to have its structure determined by X-ray crystallography. Hemoglobin is a tetramer of 4 subunits that are all myoglobin-like. Myoglobin is an intracellular protein in vertebrate muscle. So its job is to deliver oxygen within tissues. It is a helix bundle, so it is made of 8 alpha helices, and you can see each of them is given a letter designation. Additionally, within those helices is the heme. And the heme is what binds to the oxygen. Let's take a little bit of a more detailed look at that heme. So heme, we call it a prosthetic group because it is not native to the protein. It is kind of added after the protein is made. You may hear the word porphyrin ring used to refer to the heme. Heme, porphyrin ring are kind of synonymous. Now shown at the bottom here is a stick image of what the heme looks like in the protein. So here's the heme, and we can see it is bound to its 2 ligands. One is a histidine from the protein, and of course the other thing the heme binds to is good old oxygen. On the top right of the slide, you can see a more detailed atomic level drawing of the heme. And again, you can see that the iron is bound to 2 ligands. The first is histidine, and the second is oxygen. In that heme molecule, you can see that the central iron is coordinated to many nitrogen atoms within the porphyrin ring, right? 1, 2, 3, 4, in addition to the histidine and the oxygen. I want to make a note that this iron 2, is not oxidized, so it does not become iron 3 when it binds oxygen. It just becomes oxygenated, and that's why when it is bound to oxygen, we see the scarlet color of blood. When it releases oxygen, blood changes color to a more dark purple. Now, if you study the picture of the heme, you can see it contains many double bonds. It is a conjugated system, so we would expect this heme to be fairly flat since it's comprised of sp2 hybridized carbons that we know should have that kind of trigonoplanar arrangement. Let's talk a little bit now about myoglobin binding oxygen and its binding curve. So myoglobin's binding curve looks like this. It has a kind of hyperbolic shape. Now let's study the different aspects of this graph real quick, okay? We'll start with the y-axis. So YO2 is the fractional saturation of myoglobin. We can think about how much of the myoglobin is bound to oxygen. Now on the x-axis, we have PO2, the partial pressure of oxygen. We can think of this as how much O2 in the environment. And if you study the curve, you can see that as we go from less to more oxygen, myoglobin very quickly seems to bind. I would say within the first 10 torr on this graph, we see 80% of the myoglobin is bound, which means that myoglobin must be pretty good at binding. But there is a constant that we can use to understand how well these proteins bind oxygen, and it is K, the dissociation constant. So the dissociation constant is the partial pressure of oxygen at 1/2 maximal saturation. And we think of this as how well the protein binds oxygen. And let's see what the K value is for myoglobin. So first, let's find 1/2 of YO2. Here is 0.5. So we'll bring that over to the curve. And now K is the partial pressure of oxygen at 1/2 saturation. So we need to drop that down to the x-axis to find the partial pressure. And so the K value of myoglobin is about 3 torr. And so this is fairly small, which should indicate to us that myoglobin binds oxygen very well. Next up, hemoglobin.